My name is Kai Simons, and I come from the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden. And now we'll give 3 iBio seminars. They will all be about lipids as organizers in cell membranes. Cell membranes, they are two-dimensional dissolutions of oriented lipids and proteins. The matrix is made of the cell lipid bilayer. Cell membranes are made out of lipids. The matrix is made from the lipids. And then there are proteins embedded in the bilayer. Now, in the last 2 or 3 decades, the proteins have reached most of the attention. 1/3 of the genome, of our genome, encodes 4 membrane proteins embedded in the bilayer. And many proteins, other proteins, spend part of their lives on either side of the membrane interacting with the membranes. So many of the functions in our cells are membrane localized. Now, the lipids have more and more come into the background. But in fact, of course, they are as important. Membranes are made out of proteins and lipids. So take the lipids. We have the glycerolipids, sphingolipids, and the sterols. And these, if you look at them more closely, then take the glycerolipids. They are very complex. So you have the glycerol molecule, you have the head group, and then you have the 2 fatty acids. So over 1,000 individual lipid species can be analyzed. And we look at this complexity, we see that there are several head groups. There are these fatty acids in SN1 and SN2 position. They are of different length. They have different numbers of double bonds. And they can also be linked to the glycerol with ester or alkyl ether or alkyl ether linkages. So this makes this whole complexity. Also, the sphingolipids are complex. They are not based on glycerol. They are ceramide-based. We have a sphingosine with a fatty acid amide bonded to the sphingosine. This is the ceramide, the hydrophobic backbone of these lipids. And then we have head groups, phosphocholine, like we have in phosphatidylcholine and the glycerolipids, but also many glycan head groups, like you see here, the sulfatide, the ganglioside GM3, or the Forssman glycolipid. Now, the big problem has been how to analyze this diversity. So many labs still use very simple methodology, DLC, thin layer chromatography, or liquid chromatographic methods, and they can only separate the classes like sphingomyelin, phosphatidylcholine, or phosphatidylanolamine. But in fact, now with mass spectrometry, we can start to look at the full diversity of the lipids. So in the 1990s, triple quadrupole mass spectrometers came into play. There we can analyze lipid species, and we have there then the lipid species. Just means that we can't differentiate between the fatty acids. But now with the new generation of hybrid mass spectrometers, we can look at molecular species, meaning we are analyzing the full diversity. So in our institute, we have with Andrei Cherchenko been developing a shotgun lipidomics platform, which now can analyze the full lipidome of cells. The sample has to be extracted. We add internal lipid standards. We infuse them into the mass spectrometer, and then there are software programs to quantitatively analyze the lipid profiles of cells and tissues and organelles. Now, if you look at the lipid quantification, then you see here you get all these peaks, and we add the standard, internal standards, and in this way we get quantitative lipidomes, as you will see in these lectures. So, cellular lipidomes may contain up to 7,000, we don't really know, lipid species. It's like the genome. We thought first that there were 100,000 genes, and today, it's only 1/4 of that number. So the full number of lipid species are not yet known. And lipids are important. And the question is, what is this complexity good for? Why do we need all these lipids? Why do cells have to synthesize so many lipid species, metabolize them, and regulate them? Well, lipids are important in forming the shapes of membranes, and therefore they are important for cellular architecture. They are also important in signaling. You can hydrolyze away parts of lipids, and then they signal into the cell, are part of the whole signal transduction system that cells have at their disposal. They also regulate the function of membrane proteins. Protein-lipid interactions is a big forthcoming area which needs a lot of work. Now you have also the question of how these lipids are distributed in the cell, membrane trafficking, for instance, or the issue of how lipids are part of mechanisms to create subcompartments in cells. So I will now, in my 3 iBio seminars, in the first one, this one, talk about the role of lipids in organizing the biosynthetic pathway from the endoplasmic reticulum to the cell surface. Second one will be about this membrane organizing principle, which we call lipid rafts. And the third iBio seminar will be on the biogenesis of the glycolipid-rich apical cell surface membrane in epithelial cells. So, you know, proteins are made in the cytosol in cells, and then they have to be distributed to different destinations where they carry out their functions in cells. And there's also a very important pathway starting in the endoplasmic reticulum, over the Golgi, endosomes, cell surface, or for export out of the cell. Also, lipids have to be distributed. So many of the lipids are made in the endoplasmic reticulum, and then they move from there to different destinations, either by membrane trafficking mechanisms or by other means, and then we get the full lipid complement, which is different in the different membranes surrounding the organelles in our cells. So if you look now at the 3 major groups, the sphingolipid family, the glycerophospholipids, and also cholesterol. Then cholesterol. Let's start with cholesterol. So cholesterol is made in the endoplasmic reticulum. But the concentration there is kept very low. Cholesterol after synthesis is moved out towards the Golgi and to the plasma membrane and to other organelles, and there is a gradient, low in the endoplasmic reticulum, low in the, a bit higher in the Golgi, and then highest in the plasma membrane. So cholesterol is synthesized in the ER and then moved out. Now, what are the functions of cholesterol? So first of all, cholesterol makes the bilayer more impermeable. It's a more robust membrane. Therefore, you have more in the plasma membrane, which is protecting the cell. Another function is that it thickens the bilayer. The bilayer thickness increases somewhat with cholesterol in there. Now, one important question in this field is that you have now the bilayer with the lipids, and then you have the proteins embedded there. So if you look at the proteins, the parts that are facing the bilayer are all hydrophobic. And the parts, let's say in the alpha helix here, it's a transmembrane domain of hydrophobic amino acids. And very important is that the length of these hydrophobic domains have to match the thickness of the bilayer. If hydrophobic areas are open to the outside aqueous solution, that is not tolerated. So therefore, the bilayer can extend, as you see here, order, become thicker to accommodate the transmembrane domain, or the membrane domain can tilt, the protein can tilt, or it can be compressed to be covering the hydrophobic domain of the protein. Now, very important is that cholesterol-depleted membranes, like the endoplasmic reticulum, they are deformable, and therefore they tolerate transmembrane domains of different lengths. But when we increase the cholesterol concentration, then this mismatching becomes a problem. And it's much more difficult to accommodate proteins of different lengths in a cholesterol-rich membrane. This means that cholesterol induces protein sorting. So if you have a bilayer that can accommodate these with cholesterol in them, then the short transmembrane domains will come together and be separated from the long ones. And this is a principle that is used now in the trafficking from the endoplasmic reticulum over the Golgi to the plasma membrane. So the membrane is thickened towards the cell surface, which is the thickest, and this induces now a sorting principle where you start now in the endoplasmic reticulum. You put in proteins of different lengths. Endoplasmic reticulum membrane tolerates that. Then they come to the Golgi, where the cholesterol increases. And then we see the following phenomenon. Plasma membrane proteins that have to be transported further are longer in the transmembrane domains, 20 amino acids about, whereas the Golgi ones, as you see, are shorter. So you have a biophysical principle of how you sort these proteins this way. And this was work by Mark Bretcher and Sean Monroe in the early 90s. And it has now been confirmed in many ways. Now, thus we have a biophysical principle, transmembrane domain length, cholesterol increasing in concentration, we get a sorting principle. But that, of course, is not enough. So let's now go to the sphingolipids. So the sphingolipids are made in the Golgi mainly. So the endoplasmic reticulum is very low in sphingolipids, that is cholesterol and glycerophospholipids, and the sphingolipids start to be made, sphingomyelin and the glycolipids are made in the Golgi, and then increase in concentration like cholesterol towards the plasma membrane. And here comes another function of cholesterol, and that is that cholesterol and sphingolipids like each other. They associate with each other. And this leads to this principle of sphingolipid-cholesterol rafts. So in the bilayer, if we have these red lipids, ceramide-based sphingomyelin, or the glycolipids, then you see that these lipids, if you look at their hydrocarbon chains, they're all saturated. They are straight. Whereas the glycerolipids have unsaturated fatty acids, usually in the SN2 position. They are kinked. And so cholesterol likes to be under the head group of the sphingolipids, associate with each other, and pack more tightly, segregate away from the unsaturated glycerolipids. Now, this all happens in the outer leaflet of the bilayer, the outer leaflet facing the outside if it's the plasma membrane. Now, also the inside of this assembly is more ordered, and here we don't know exactly what is the principle here, but they come together and form these liquid ordered, more tightly packed domains. And important here is this, that you see here the sphingomyelin and the glycosylation are more saturated, as I said. The fatty acid is longer, and they have hydroxyl groups, so they can intermolecularly hydrogen bond. And this assembly also becomes somewhat thicker. So this now is a principle for segregation in membranes. And rafts function as a sorting principle because they not only contain lipids, the sphingolipids and cholesterol and the others, they also contain proteins, specific classes of proteins. We have the GPI-anchored proteins, glycophosphatidylinosyl anchored, 2 saturated fatty acids sticking into the outer leaflet of the bilayer, or we have on the inside doubly acylated proteins that have 2, a myristylate in the amino terminus and then a palmitoyl nearby, and they also associate with these. And then we have transmembrane proteins which also like to be in rafts, and many other proteins that are excluded from the rafts. So rafts then come in as a sorting principle in the trans-Golgi. Sphingolipids, cholesterol, proteins are sorted and then transported to the cell surface. Now, let's look at this in more detail. And we shift now to yeast, Saccharomyces cerevisiae. Very great model system for cell biology because it's simpler than most other cells. So there are two pathways from the trans-Golgi network in the Golgi, the exit station. One, the red one you see here, going to the surface, and then another one going to endosomes, and then to the surface. And the idea is now this red pathway, is it a raft pathway? If we really think that these rafts are involved in the sorting process, then we have to get some evidence for this. So the carriers that are formed in the trans-Golgi network that you see here, if we would isolate them, they should be enriched in sphingolipids and sterols and have much more than the donor compartment than the trans-Golgi network. So let's look at this. So now we want to isolate using immunoisolation techniques. Yeast secretory vesicles coming from the Golgi and carrying a raft transmembrane protein, which is called this fused mid cargo protein. So now the idea is to isolate the vesicles formed in the Golgi carrying the fused mid cargo. Okay, yeast is great because there are genetical strategies. Randy Sheckman is talking about these in his iBio seminar. There are mutants for each step in the pathway, and you can use these to study questions of this sort. And there are mutants, for instance, the Sex6 mutants, which block the delivery of these carrier vesicles on the way to the surface. They accumulate in the cytosol. And now we express this cargo protein, the fused mid cargo protein, and then we restrict the temperature, we lower the temperature, these are temperature-sensitive mutants, and then we get this accumulation, as you see, of these red vesicles in the Golgi. And now, coming from the Golgi, accumulating in the cytosol. And if you look at them, then one subset of these red vesicles is those that we want to isolate now. Okay, so we homogenize the cells after we have accumulated the vesicles in the yeast cells, and then we fractionate, and the final fractionation is a cell density gradient centrifugation, and then we take the fraction in the gradient that contains our fused mid transmembrane cargo protein. Now look at it. This fused mid protein has a DFP tag, so we can see it, color. It has a TEF cleavage site, so you see, and it has 9 MUC epitopes. It's epitope tagged. Now we have this fraction. We then add antibodies against the MUC epitopes. They bind to it. And now we take a cellulose fiber immunoadsorbent, which contains antibodies against the MUC antibodies. And then we pull down the vesicles. And when we have done that, we now cleave off the vesicles using this TEF protease. And what we get? We get vesicles, which look exactly like those that accumulated in the cell, 100 nanometer diameter. And now we want to analyze the lipid composition of these immunoisolated, very pure vesicles carrying our raft cargo protein. Okay, so the yeast lipids are similar to mammalian lipids. They have sphingolipids, ceramide-based, but the head groups are a bit different. They have a inositol phosphate, that's the simplest one, and you can add a mannose, or you can add even one more inositol phosphate. So we shorten this with IPC, MIPC, and MIP2C. It's only 3 classes of sphingolipids. They don't have cholesterol. Instead, they have ergosterol, which is very similar. So these are the lipids we are looking at now. We look at the whole lipidome using our quantitative lipidomics platform that we have developed together with Andrei Chechenko's group. And now we can just extract the yeast vesicles that we have immunoisolated. We add the internal standards. We infuse them into the mass spectrometers, and then we analyze. Now, we are now analyzing the molecular lipid species using hybrid mass spectrometers. And if we look at the whole lipidome, we see that there are 200 molecular species consisting of 17 lipid classes. And now let's look at the vesicles. We have also isolated the donor compartment, the trans-Golgi network, and now we compare them. And what we see here is the full spectrum here. And if you look at the ergosterol is the major component in the vesicles, and the other major component is down there, the MIP2C, the most complex sphingolipid. So the vesicles are enriched exactly in those lipids that we hoped they would be enriched for. And then you look here, you see that sphingolipids and ergosterol are over twofold enriched over the trans-Golgi network isolates. So, indeed, we have now shown that this red pathway is a raft pathway because we have ergosterol and sphingolipids enriched in these fused meat vesicles. So we have seen lipid sorting. So in the process of making these vesicles, we get more sphingolipids and more ergosterol in there. Now, we also use a visual screen to look for genetic mutants which are somehow blocked in this pathway. And the problem is this. Well, first of all, we use the same cargo protein, the fused mid GFP, but only containing GFP and not the rest, not the MYC epitopes. And important in this case is that there are two pathways to the surface from the Golgi. Now, if you block one pathway, it turns out that the components which usually take this pathway take the other pathway. It's like going to the station from here. The road down the hill is blocked. Then we take the other road and we reach a station. It takes a little longer. And if there's a real problem, if this is in a city, we have a traffic jam. But the traffic jam will then, in the end, get the cars where they should. This is the same thing in the cell. We use this traffic jam as a screening principle. That means now that in the wild type, not in the mutant cells, we are deleting all genes in yeast and looking at each deleted mutant. And in the wild type, we see the protein reaching, the way we do the assay, the cell surface, as you can see here. But in the traffic jam mutant, you see inside accumulation. That is the traffic jam. Well, looking at all the genes, we got 24 mutants, and very important, we got 6 lipid metabolism mutants. And what were they? Well, they were 2 ergosterol mutants in the ergosterol synthesis pathway and 4 sphingolipid mutants, exactly those lipids that we have now implicated in this transport pathway. So if you look now at one of these, for instance, SUR2, it's a hydroxylation mutant, and this leads to mis-sorting to the vacuole and to a Golgi block. And another mutant is LO3. It's a fatty acid elongation mutant and leads to mis-sorting to the vacuole. So small changes in lipid structure leads to traffic defects. So in this case, if you look here, you see these hydroxyl groups are missing in the hydroxylation mutant. And if you look at the LO3 mutant, you see down here, it's not a C26 long fatty acid, it's a C22, it's shorter. And if you go now to the raft concept, what does it mean? Yes, less hydroxylation means less intermolecular hydrogen bonding. So you don't have this association between the lipids in the assembly functioning as well. And the long saturated fatty acid probably penetrates from the outer leaflet into the inner leaflet and organizes it in this way. Now it's shorter and it's not doing its job properly. So you can see that these changes indeed lead to defects as will be predicted if rafts are involved. So now another prediction is that when you take the vesicles that are formed in the Golgi for delivery to the cell surface, if they are containing rafts, the packing of the rafts should be higher. That's also one of the issues, as you remember. Now, how can we measure that? Well, we can take a probe. It's called C-Lordan. It goes into the membrane, and it can feel how much water is in the membrane. So if the membrane is disordered and not so tightly packed, there's some water molecules in the membrane. But if it's more tightly packed, there is less water. And you excite then you take, for instance, these vesicles, and you add the C-Lordan, and now you excite at 800 nanometers, and then you read the spectrum. And if you have high lipid packing, the emission will be at 430 nanometers, and if you have low lipid packing, it's 490 nanometers. And if you do this, then we see that the vesicles are more tightly packed than the donor membrane, the TDN. Again, speaking for a liquid ordered change to a more packing, higher packing during this formation of the vesicle. So we have now shown that, gone through with you some principles where lipids are involved in membrane transport, trafficking from the endoplasmic reticulum to the Golgi, and from there to the cell surface. And we have seen that cholesterol leads to a sorting of proteins depending on their transmembrane domain length. Short proteins stay in the Golgi, and longer proteins, longer transmembrane segments go to the plasma membrane. And then comes this principle of rafts into play, where sphingolipids and sterols are sorted with proteins to the cell surface. Now, I must, of course, not forget to mention that there are, of course, other principles involved as well, but we are not going through them here. For instance, the major principle of making a vesicle from a donor compartment for delivery to the next compartment is by having transport signals in the cytosolic domains of the cargo protein, which then bind to adapters, cytosolic adapters, which in turn bind to coats, like clathrin coats, and in this way you form with other proteins a vesicle which then takes off. And we are here thinking about another model where lipids come into play, where the raft concept leads to sorting of both lipids and proteins. And in the final lecture, I will come back to this issue, how this mechanism works. And here I'm going to end. And the next lecture will be on this dynamic compartmentalization concept, the raft concept, how it works in more detail. And finally, some acknowledgement of the people who were involved in the recent studies in my lab. Thank you.